Well, good afternoon, everyone, and welcome to this, the latest in our series of online SCItalks. It's very appropriate time for today's talk because you may or may not know, but this is actually World Antimicrobial Awareness Week, which is a global initiative to tackle the growing problem of bacterial antibiotic resistance, which I've seen recently described as another pandemic that threatens our whole healthcare system. So today, the chairperson is me, Peter Hambleton, and we're being helped by Bryony Parker, who will deal with all the questions. If you do have questions, please use the message typing board on the left or right hand side of the screen to submit your questions to Professor Sockett. Professor Sockett's team are evaluating the Bdellovibrio predator strains as possible anti-pathogen agents and working to understand their mechanisms for gram-negative bacterial killing. And these are, in fact, very multifactorial. About our speaker today, Professor Liz Sockett is a bacteriologist who researches and teaches at Nottingham University. She has a considerable list of prestigious affiliations. She's a member of the Microbiology Society, a fellow of the American Academy of Microbiology, and a fellow of our own Royal Society. And her research group is looking at the predatory bacterium Bdellovibrio bacteriovorus, with funding from the Wellcome Trust, from DARPA, BBSRC and the Leverhulme Trust. This bacterium naturally kills gram-negative bacteria, and Professor Sockett's team are looking now at the potential for these predatory bacteria to be used in the fight against antimicrobial resistance in gram-negative pathogens. So over to you, Liz.
Thank you very much. So it's very nice to be with you today, and I'm going to tell you about predatory bacteria. On the left-hand side of the screen, you can see an actual electron microscope picture of a predatory bacterium attached to another bacterium. And on the right-hand side, you can see a cartoon of the same process. In my laboratory study, the yellow predatory bacterium that you can see on the right. It kills a diversity of other bacteria, whether they are or are not resistant to conventional antibiotics, and some penicillin resistance is shown in the center of the slide. So our laboratory works on antibiotic-resistant bacteria from QMC, which is our local NHS hospital. We specialize in the predatory bacteria and trying to see how they can kill those pathogens despite the antibiotic resistance. And as Peter said in the introduction, a variety of different funders have, if you like, prodded and funded us into looking into these organisms. And today it's a big team effort, but I will be telling you about these wonderful people and what they have discovered. So infectious microorganisms are being selected for antibiotic resistance by use of the very treatments that used to cure us. And they're alive, evolving and adaptable. And Sally Davis, the previous chief medical officer, said that they really should be added to the National Risk Register of Civil Emergencies. And as we're in a pandemic now, I think we know very well what different microorganisms can do. She's very concerned that antibiotic-resistant infections that are resistant to the conventional medicines that we've become used to would form a real problem for the future. And just because, sadly, we are in a viral pandemic at the moment, this doesn't mean that this threat has gone away. So we want to apply the knowledge from how bacteria naturally kill each other. And I am a research scientist and an academic. I am not yet a business person or in any way a pharmacist. But I use science and I use the knowledge of bacteria in a way for human good. So this is the bacterium that we study, discovered by chance, like all good science, in Germany by Heinz Stolp in 1962, found in soil originally, but these bacteria are found in water, soil, oceans, ponds, all over the world. They have evolved to dine privately inside other bacteria, and this is their natural lifestyle. And Heinz Stolp was growing some gram-negative bacteria, which we'll talk about later, on a plate, And he found that some agent was making holes in those bacterial lawns that he was growing in his laboratory. So a little bit like the Alexander Fleming situation with the discovery of penicillin. And so what he had actually found was ultra tiny bacteria that could invade inside and live within other bacteria, killing them. Now, this private dining that these bacteria have evolved to do is a particularly bacterial process, but actually represents processes that the pharmaceutical industry does not have, and that these organisms know how to do, but humans do not. And the great thing about eating bacteria from the inside is that you reduce some of their immunogenic core materials. So materials that may interact with the human body and cause an inflammatory response or even a lethal response. So these Bdellovibrio bacteria are natural killers, and our approach has been to link with the NHS. Here are some staff members from NHS Public Health England before the pandemic, and they would supply us with bacteria that had become problematic in a hospital setting. Now, many of these bacteria are known as escape pathogens because they escape the actions of antibiotics. They include these bacteria here, such as Klebsiella and Acinetobacter, and maybe Escherichia may be more famous, and people may have heard of that bacterium. So these organisms are causing problematic infections in the NHS, and people are dying of these infections because those bacteria have become resistant to conventional antibiotics. Now, what we have in this organism, our Bdellovibrio bacteriovorus, is a novel bacterium that is not itself a pathogen. What it is an organism that delivers many antibacterial factors during its invasion process into other bacteria. It carries out this lifestyle where it collides with and attaches to another bacterium and invades into its outer compartment, a position called the periplasm. It then kills the bacterium at that stage, but keeps it intact for another 3 hours. And it systematically uses a series of enzymes to dismantle that bacterium and use it to make more of the Bdellovibrio. So in this way, the Bdellovibrio is converting a harmful pathogen to harmless Bdellovibrio bacteria. And we will see from our studies a bit later in the talk how harmless, because the Bdellovibrio are very lethal to other bacteria, but they are not lethal to humans. They're also not lethal to animals. And although human research is in its infancy, there has been occasions of Bdellovibrio being isolated from the guts of young children, for example, where they're dwelling as part of the natural microbiota of a children's gut contents. We haven't yet done a lot of trials in human susceptibility to Bdellovibrio, but we have done some trials in animals, which I'm going to talk to you about. Now, half of our laboratory ask about these predatory functions, because these Bdellovibrio bacteria have got things that are unknown to pharmaceutical science, and they are a series of enzymes and adhesins which allow them to do this attachment and invasion into other bacteria. So half our group study those functions. The other half look at the whole live predatory bacteria and see them as treatments. And so the talk will take 2 parts. So I want to show you this process in action at the moment. The Bdellovibrio bacteria can attach to the outside of the bacteria and invade them. And I'm going to show you a few seconds of a movie, which is 4 hours of that process speeded up. So when I start the movie, you will see a Bdellovibrio enter from the bottom side of this slide and attack this bacterium at the bottom, just here. Next door is a bacterium that's already been invaded by Bdellovibrio, and you will note it has rounded up. All the other bacteria on the slide are rod-shaped, which is their original shape before invasion. So let's start that movie and see what happens. So here comes the Bdellovibrio and invades the first bacterium. It rounds it up and has killed it. They're now inside consuming the contents of that bacterium and the neighboring one, and you will see them break out when they have done that. And 4 hours has passed, and out come the Bdellovibrio. They can glide across the surface, and they can seek out, collide with, and repeat the process, invading and killing more bacteria. This whole process takes 4 hours, but it is a process that kills bacteria, replicates the Bdellovibrio, and moves them on to do more. You can also see this in the top right, where the bacteria are mobilized, and this time the Bdellovibrio are in a liquid medium, and they're coming like torpedoes to kill these bacteria. So you see them attach at the top, they pause, and then they use a dragging mechanism to drag themselves through a pore that they have made in the outer surface of the bacteria and enter them. They do all of this naturally, and that invasion process takes about 15 minutes. They round up and kill the bacteria early and then digest them sequentially. Now, this is an amazing process, and in 2017, we were able to work with a very talented PhD student called Erkin Kuru in the USA to actually visualize what was happening at the outer surface of a bacterium invaded by a Bdellovibrio. We used fluorescent D amino acids to label our Bdellovibrio red, and we chased in some free fluorescent D amino acids to ask what enzymes could be using amino acids and modifying the bacteria. And indeed, was there a pore being formed through which the Bdellovibrio could enter the prey? The answer is definitely yes. And you can see those blue D amino acids lighting up a porthole, a molecular porthole that these bacteria assemble and drill into the wall of the bacterium they're invading. They go through the outer membrane of the bacterium and then build this wall and enter through to the inside of the wall of the bacteria they invade. This is science that is unknown pharmaceutically, but is all taken together as a concerted effort by these bacteria. And so some of our purpose is to understand these processes. So they build a porthole and enter the bacteria. As they do so, they round up the bacteria and they change them from a rod shape to a rounded shape. And this is actually a form of reinforcement, preventing those dead bacteria from bursting so that the Bdellovibrio can consume them. The Bdellovibrio then close up the porthole and attach a filler to the porthole and live within the bacterium, which is now dead, and consume it. And indeed, that reinforcement is an amazing process, a little akin to human painting of walls inside baptistries. They actually apply these amino acids to make strong bacterial cells, which are dead, in which they can feed on the contents of the formerly live bacterium. Novel enzymes are doing this, and then a series of hydrolytic enzymes digest the prey bacterium. And one of the great things about this Bdellovibrio predation process is that a bacterium made of certain biomolecules is able to organizedly kill another bacterium made of the same type of molecules without killing itself. Now, Bdellovibrio can do this because it secretes novel enzymes that are predatory into the prey bacteria, and some of them begin with endopeptidase and D-amino acid changes, as I have shown you. There are also deacetylase enzymes, and all of these work uniquely on the cell wall of the bacteria. We also find that that modification that the Bdellovibrio do can actually ordain that they can break through the cell walls at the end of predation and escape. And if anyone is interested in that, I haven't made this a major part of my talk today. I'm concentrating on the whole bacteria, but I'm also very happy to answer questions about the enzymes because we do a lot of work on this. Now, as I said, the Bdellovibrio really do consume the bacteria that they go inside. This is very different to bacteriophage therapy, which bursts bacteria quite early on and releases quite a lot of the material that those bacteria were made of. So with a pathogen, you sometimes have pathogen-associated molecular patterns, which are molecules that can be harmful to the host. If, however, you have an indwelling bacterium that doesn't have those, but is consuming the pathogen, you will convert the material of the pathogen to harmless Bdellovibrio. And you can see that in this movie here, where the Bdellovibrio has invaded a fluorescently tagged E. coli bacterium. The black Bdellovibrio is consuming the E. coli material, is making more Bdellovibrio, and indeed 5 are formed in this particular cell. And then the cell is finally burst, and the PAMP material is really reduced. So this is the big difference between using a conventional antibiotic or a bacteriophage. The Bdellovibrio actually reduces down the material that even the pathogen is made of. So not a lot of dead pathogen is left. So this natural life cycle of predation has many features in it that eventually kill the pathogen and will replicate more Bdellovibrio. And we will look at their activity inside living animals in a moment. Now, I want to mention briefly that I have a great collaborator, Dr. Andrew Lovering at University of Birmingham. And together we do work on the mechanisms of these predatory enzymes. We really love our science, and this quote that is at the Harnack House in Berlin really sums it up. We feel that we have researched the researchable and indeed made more things researchable in these bacteria. So together with Andy, we actually work on enzymes that can allow bacteria to enter others, to modify those bacteria, to close up a pore or make a pore, to sequentially break down the materials inside a bacterium, to not lyse themselves, but then finally to exit out of those bacteria and find new prey. And together with Andy, we are in a world of like novel bacterially produced pharmaceuticals. We study them as interesting natural products, but they really are agents of killing bacteria that are unwanted. Now, currently on our Wellcome Trust Investigator project, we are defining the enzymes of the porthole production process. So we are looking at that porthole that you see outlined in blue that the bacteria make in the surface, and we are looking at its connection through to a porthole lower down that is in the cell wall of these bacteria. And so we are working hard right now on working out all of the different types of enzymes and attachment processes that allow these bacteria to cross through a living organism into their insides and then start to consume them. Now, that process itself is complex, multifactorial, and hard to resist. And when you see it in whole living bacteria, it is really therapeutic. Now, in earlier work, one of our students, Emma Saxon, looked at Bdellovibrio on a living surface, killing other bacteria. Here we see a very simple living surface. It's a mushroom that is suffering from a pathogen, which is a Pseudomonas called Pseudomonas thalassii. Here is the mushroom surface in gray and the pathogen in yellow. And you can see the Bdellovibrio in blue attaching to and starting to invade that mushroom pathogen. Now, if the pathogen is left to its own devices, it produces a brown rot on mushrooms. But if you apply the Bdellovibrio to the pathogen, the brown rot does not develop on the mushrooms and you're left with a watermark. So that's an interesting property of these Bdellovibrio bacteria in a very simple, innocent pathogen relationship with an innocent mushroom. We have also, working with Rob Atterbury and Laura Hobley, who both have lectureships at the Vet School in Biosciences on another campus of Nottingham University, investigated also what happens if we give drinks of Bdellovibrio to young chicks. We gave drinks of Bdellovibrio to birds that had no other infection, and we found that the birds were well and healthy and had no difference in their growth and feeding. But if those birds had been pre-inoculated with a salmonella infection, we found that the Bdellovibrio could reduce the salmonella in the bird's guts. And indeed, if we dissected out the bird's guts and looked at them, we found that birds that were treated with the salmonella had quite inflamed guts, a lot of white blood cells in there. But if treated with the Bdellovibrio, the gut appearance was reduced back to the non-inflamed version. Now, the Bdellovibrio was short-lived in the guts of the salmonella, and indeed probably did one round of replication and killing of the salmonella pathogens inside the bird's guts. But indeed that was enough to reduce the numbers of the salmonella that were present. So at this point, we had shown that you could alter inside a living creature, at least in the gut, the levels of a pathogen using live Bdellovibrio. Now, at this point, we got nudged by DARPA, and in the shape of Barry Palotta, who's a program director of one of DARPA's programs. And he said that he'd seen some of the movies of predation in one of our papers, and that DARPA was starting a potential therapeutics testing program with Bdellovibrio. And so we should think about doing that work. And so we applied and began a new chapter with several other laboratories in the US and also in Korea and Israel working on this area. Now, for us, we were not able to dive into humans to use as our patients. And indeed, we're still not working in humans yet, but we had to gather evidence of a much more controlled, injected application of Bdellovibrio. And to do this, we met Serge Mostovy and Alex Willis. And Serge works at Imperial College London at the time that we met him, and now works at the London School of Hygiene and Tropical Medicine. Alex I actually knew from earlier, but she was his PhD student. She'd been one of my undergrads at Nottingham. So it was lovely to meet up with them again. Now, Serge's laboratory was known for using unusual patients to test infections. And instead of using patients like a human, they're able to use a miniature patient, which is a transparent zebrafish larva. And the zebrafish larvae have an innate immune system, very like that of humans. It's a surprising thought. Zebrafish will lay a large amount of eggs that will be fertilized, and you can actually study the larvae in large numbers. You can anesthetize them, and as Serge had shown, you can treat them with a pathogenic infection and see the outcomes. So we were able to use these small patients in a zebrafish infection model. So Serge had already shown that Shigella flexneri, which is a pathogen, can cause infections when injected into the zebrafish larvae. And because they're transparent, and if you make the Shigella fluorescent, you can actually look at the back of the head of the larva to see how that infection is going on. And you can study the growth of the green fluorescent pathogen. You can also sacrifice humanely the fish, and you can count the number of live bacteria that are in them by a colony count. You can also inject a large number of these zebrafish larvae humanely, and you can test for their survival if injected with a pathogen and just a buffer control. You're also able to immunomodulate the fish and make them produce different amounts of neutrophils and macrophages, the major cells of the innate immune system. And indeed, you can get transgenic fish where you can see where those white blood cells are in the fish because you can color them transgenically. So Serge's group had set all of this work up with some other collaborators and worked on it and knew how many pathogens it took to kill zebrafish larvae. We were able to add another component to this experiment by using red fluorescent Bdellovibrio bacteriovorus and asking what happens if you apply Bdellovibrio that are red labeled so you can track them and see whether you can use them as a treatment. So the first experiments we did, we took our Bdellovibrio and labeled them red and injected them into the hindbrain of the fish. This was in parallel experiments to repeats of Serge's work, where the green fluorescently labeled Shigella were injected into the hindbrain of the fish. Now, Serge had already shown that a lethal infection could be produced by a certain number of Shigella cells injected into the hindbrain of the zebrafish larvae. And at first, we just injected Bdellovibrio alone to check whether or not they were pathogenic, because the idea of injecting bacteria into the brain of a living creature to act as a treatment is somewhat extreme. Now, I already told you that Bdellovibrio are not pathogenic to animal cells. And indeed, we found that over a 48-hour period, even with high numbers of Bdellovibrio injected into the hindbrain of these fish, they were perfectly well. This was in contrast to if a pathogen was injected. What we also found was that the Bdellovibrio numbers remained remarkably high for the first few hours after we injected them into the hindbrain. And that is despite the fact the hindbrain is accessed by the innate immune system. So even at 48 hours, there was still a sizable number of Bdellovibrio left alive inside the zebrafish. Next, we tested the combination. And we took those Bdellovibrio that were fluorescently labeled with red, and we took the green Shigella bacteria, and we injected first the pathogen and then the treatment of the Bdellovibrio treatment. So that was a really good thing to do, and we were able to visualize these infections as they proceeded. So what we saw over a number of hours was the growth of the Shigella if it was injected just with a buffer control. And then we were also able to monitor the effect of adding the red Bdellovibrio after the green Shigella. Let me show you some movies of this in action. So here is an immobilized, anesthetized zebrafish larva down the microscope, and you will see time running, and you will see that it's been injected with green fluorescent pathogen, and over a period of hours, the green fluorescent pathogen grows, and eventually over more hours, this will lead to the death of these fish after a period between 48 and 72 hours. Now, where we inoculated with the same pathogen, but instead of just adding buffer, we added red Bdellovibrio, the outcome is much different. And by the end of the movie, you do not see the full growth of green fluorescent pathogen as you do see where there is just the buffer. So if you watch this movie, you will see the red Bdellovibrio interact with the green pathogen, and they reduce that green levels of pathogen, and indeed themselves eventually are snuffed out, but the levels of the green pathogen disappear on the Bdellovibrio treatment. And so the outcome is very visual in single fish. We then went on to do this in multiple fish and to count all of the infections that we produced. So we had Shigella and buffer alone, which are in the open circles here. And over time on this axis, you could see the numbers of Shigella would grow in the zebrafish hindbrain treated only with buffer. If we treated with Bdellovibrio in the closed circles, you can see the numbers would drop. When we looked at animal survival, we could set up many of these infections with the anesthetized larvae, and we could check their survivability over 72 hours. So if we had the Shigella with the buffer, we saw 27% survival after 72 hours. If we had added the Bdellovibrio, 67% survival. We could use large numbers of these zebrafish larvae, and we could get good statistics on these data. Very rewardingly as well, we were able to actually visualize individual bacteria in the body of the fish. And to do this, we had to examine them a bit further down the fish where it was even thinner, but more amenable to high power microscopy. And we could look inside and see individual rod-shaped pathogens being converted to that typical rounded shape I showed you earlier in the movies and invaded by the Bdellovibrio bacteria. So it was the actual predation that was killing these pathogens inside the live zebrafish. Now, we also found that there was stimulation of the innate immune system during the killing process by Bdellovibrio. And we found that the Bdellovibrio were actually killing the Shigella, but also stimulating the fish immune system to join in with that killing. Now, we knew that this was happening because we were able to modulate the production of innate immune cells by using something called a Pu.1 morpholino. We found that macrophages and neutrophils are reduced, as is well known, if we treated with this Pu.1 morpholino, and we could make fish that had less macrophages and few neutrophils. Now, when we gave those fish an infection, they did quite badly because they had no innate immune system, and indeed at 72 hours, their survival was 0. If, however, we added Bdellovibrio alone to fish without those innate immune cells, we got 25% survival. In our control experiment, where we used a morpholino that was scrambled as a control, we saw 67% survival on Bdellovibrio treatment and 32% survival if we just had the Shigella present. So there is an additive effect of Bdellovibrio predation and the immune system giving the overall effect. Yet without Bdellovibrio, the immune system is not noticing the Shigella and is not killing the Shigella in sufficient quantities to prevent the infection. So a summary so far is that immune cells work with the Bdellovibrio to control the Shigella in vivo. There is an effect of predation, but there is also an effect of stimulating the immune system to join in the clearance of the Shigella from the fish. Now, we have been evaluating those leukocyte responses, the neutrophils and macrophages. And something you can do is watch them in transgenic fish. So here the colors are different. The Bdellovibrio is in blue and we inject it in here. We have green labeled neutrophils and red labeled macrophages. And what we see is that there is movement of the macrophages and neutrophils, but it's not actually stimulated massively by the Bdellovibrio. And indeed, if we follow that movement, we don't see a big difference in the actual movement of the white blood cells, whether there is a pathogen present or whether there is a Bdellovibrio there. What we have discovered, though, is that the Bdellovibrio is actually signaling different activities in the macrophages and the rest of the fish. And that is something that we're actively working on at the moment. We found that the Bdellovibrio interact with the white blood cells at the injected fish hindbrain. Now, indeed, we went on then to study cultured human white blood cells. So we used human U937 macrophage-like cells, and we found that they would take up Bdellovibrio. Now, this is not invasion of those cells by Bdellovibrio. this is engulfment of those cells by the macrophages. The macrophages take up these little green fluorescent Bdellovibrio, and here's the red human macrophage, and inside are the green fluorescently labeled Bdellovibrio. And indeed, just like in the fish, the Bdellovibrio stay alive for up to 48 hours inside the macrophages, but are then slowly but eventually cleared. Now, this is remarkable, but what is also good is that the human macrophage cells are not damaged when harboring those Bdellovibrio bacteriovorus. And we counted the numbers of engulfed Bdellovibrio and were able to show that there was no effect on the viability of the macrophages. Now, this supported work by other laboratories as well that show that where Bdellovibrio might be engulfed by some human cells, they do not kill them. And so Bdellovibrio has a lifetime where it is quietly and apparently harmlessly sitting inside a human white blood cell. But it can stimulate that human white blood cell to do more killing of other bacteria that are pathogenic when the Bdellovibrio are around and actively killing those bacteria. We have looked at inflammation, and here are some common inflammatory markers that white blood cells may produce. And we have looked at the levels of inflammation caused by Bdellovibrio, which are shown in green here, and compared that inflammation to that caused by pathogens. And we're showing Salmonella in blue and Klebsiella pathogens in red. Now, the Bdellovibrio consistently give a lower inflammatory response than do any pathogens. So there is not an overinflammation that is causing this effect. So we're now working on that host response, and that's the work in progress at the moment. We're working towards publishing what it is that is being sensed inside the zebrafish and why predation plus that host response kills pathogens when the host response alone was failing to do so. This is very important because when we have bacteria that are antibiotic resistant, we really need the immune system to take an extra role in killing them and to not let them multiply in the body. And it seems that the products of predation, both the interaction of the Bdellovibrio with the innate immune system and possibly production of the broken pieces of pathogen, are signaling to the host to join in and help the Bdellovibrio kill pathogens that they were failing to do so. So we now know that predation by live Bdellovibrio inside a living host can kill gram-negative pathogens. We know that the inflammatory response is low, but they do stimulate the host immune system to tackle that pathogen load it is not otherwise able to clear. We know that Bdellovibrio take up a temporary residence inside cultured human macrophages, but live dead assays indicate that those macrophages are not killed by that process, and eventually the Bdellovibrio are cleared. But this is a time window of an opportunity to treat infections and enhance that immune effect. We found also that Bdellovibrio circulating antibodies in donated healthy human sera are generally low, and that those human sera do not contain antibodies to Bdellovibrio that would wipe them out as soon as treated. This suggests that we may be able to apply a dose of Bdellovibrio bacteria in future medicine safety trials. Now, there are hurdles to overcome. Appropriate production of large-scale cultures of friendly bacteria at a pharmaceutical quality, that's a really big job to do. And as we've seen from vaccine production, though, it can be scaled up and it can be done quickly. And Dr. Jess Tyson in our laboratory has dedicated herself to that and working with Tim Overton at Chemical Engineering in Birmingham. And together, they are working with us to pilot this project. We've had to scale this back just a bit. The lab has been delayed into working in 2 shifts, delaying a little bit of this future work, but we are making progress. It's a novel therapy. Injecting bacteria into humans isn't a normal thing to do, but human and further animal trials and emergency therapeutic use might still be the route to human trials. As COVID vaccine work shows, you can speed this stuff up when you need to. So here we have some pilot production and some investigation into the parameters to produce 50 litres of Bdellovibrio on a large scale. And we're just beginning to work towards having enough large scale bacteria for bigger trials. So you've caught us at a good and interesting time. So I'm going to sum up and let you ask some questions. Bdellovibrio can certainly be our allies against bacterial infections. You can inject them. and they can become a kind of bacterial fire extinguisher. You will get one injection of these friendly bacteria, and eventually your immune system will raise antibodies and recognize those bacteria. But one injection in a compartment of the body has a lot of antibacterial killing activity. It doesn't kill the host, but it stimulates the immune system to join in. That host response is synergistic with the predators. I mentioned earlier that the work with Andy Lovering shows that the enzymes of Bdellovibrio may themselves have antibacterial properties, but they individually are much more like antibiotics, and bacteria could become resistant to them. It is the fact that Bdellovibrio make that complex porthole and put many enzymes in together that means that resistance to Bdellovibrio does not develop in other bacteria. And we've used experiments over 50 weeks of adding Bdellovibrio to prey, looking for resistance and not finding it. Now, obviously, further production, development, and testing is key, and Jess is very much involved in that. Whole Bdellovibrio aren't antibiotics, but a single injected dose may have a future role in localized infections, in the brain, in the peritoneum, maybe in joints, maybe in the bladder. And we need to do that work. It's not done now, but these are future areas. We couldn't inject Bdellovibrio directly into the bloodstream because there is a risk that you might cause strokes in the bloodstream because they are particulate in nature. But there are many other compartments where infections would be that we could use them. It's not easy, but it's fascinatingly interesting. Our projects are understanding this complex way that nature can be antibacterial. And the motto is, if you want to kill bacteria, ask a bacterium. So I'm going to let you ask in a moment, and I just want to thank these people who've been really instrumental in the Zebrafish work and in the infection work. Jess Tyson, you can see he has played a big part in this. Our funders who've been absolutely fantastic and worked with us from the very beginning 20 years ago when it seemed crazy to even study these bacteria, all the way through to now. So thank you very much. These are our collaborators, and I'm happy to answer questions.
Well, thank you very much, Liz. That was a fascinating talk. I thought, you know, in the end, you probably answered all my questions, but I was a bit puzzled as to, you know, I take your point about the fact that the invasion and the production of pores is multifactorial. Therefore, that does count against evolving resistance, but it does nevertheless, considering how long these have been around, it is surprising to me that you haven't found evidence of bacterial resistance.
So there are strategies that bacteria can employ to reduce predation, and indeed that occurs, but in a similar way to the fact that lions and zebras can coexist in the world, where the lions are the predators and the zebras are the prey. It is very similar in these bacterial relationships. And so the Bdellovibrio can have a lifestyle where they are predators, but they indeed have a second lifestyle where they grow extremely slowly between predation. And so they can enter a like dormant phase. So a bit like a well-fed lion lying down while some more zebras escape. Now, bacteria that are prey can coat themselves in outer surface layers, but that themselves will cause less fitness. And so the Bdellovibrio have to work harder on an outer surface to find a little chink in the armor to invade. But that is one of the few means by which other bacteria can reduce the Bdellovibrio effectiveness. Right, Bdellovibrio are preyed upon by viruses as well. And so they themselves can be killed by viruses in an environment, just like we can be killed by COVID, very sadly. But indeed, there are always predators for predators. But actually, it is a situation where they are very naturally able and multifactorial in their killing.
Okay, fascinating. You know, to get large-scale culture, then presumably you have to have, a bit like growing viruses, you have to infect bacteria to get to multiply the Bdellovibrio. You can't grow them independently of bacteria.
So it is possible to grow Bdellovibrio in an independent means, and there is a sort of mutant pathway whereby you can do that. You can take a derived Bdellovibrio strain and grow it host independently. For its efficacy as a predator, it is lowered if you do that, So the strategy we've taken is to use bacteria generally regarded as safe as the prey. So there are plenty of bacteria that are used in the food industry and in pharmaceutical production that are GRAS bacteria, generally regarded as safe. And so we're using them as the prey for the production of the Bdellovibrio in our pilot plants. And so that's the strategy.
Okay.
And luckily they are then predatory on all bacteria rather than all gram-negative bacteria, not just the one you grew them on.
All right. I was going to ask about that as well. They're not.
Yeah. They're not specific.
Not strain specific, but it's only gram-negative bacteria that.
Yes.
Yes. Okay. Interesting. Yeah. Again, I was thinking about the potential for human application. Presumably, it's easier to see them being used in a veterinary situation. I mean,
Sure.
Resolving the veterinary problem is part of antibiotic resistance solving.
It is,
isn't it?
definitely. Yes, it is.
Yeah.
I definitely agree.
Yeah. Brian, are there any questions from anywhere else?
Yep, so we've got quite a few questions coming in, so thanks very much, Liz. The first question is from David, who says, have you investigated what effect these agents might have on human gut bacteria?
We have done that in vitro, but we haven't done that in vivo yet. And so some other researchers are interested in looking at the gut microbiota with Bdellovibrio. We have looked at individual gut bacteria with Bdellovibrio, including Klebsiella, because Klebsiella are quite problematic bacteria because they can be asymptomatically carried in the gut, but refluxed into the lungs in patients who are hospitalized and in bed a lot. And so they can develop a lung infection where they have harmlessly carried Klebsiella, but actually removing Klebsiella would be a good idea. And there's a study in rat guts, which is being carried out by Daniel Kaduri in the USA, where he's noted the amelioration of Klebsiella in the gut of rats. At the minute, we aren't licensed to actually give the drinks to humans yet, but that will come eventually. And we have to be very cautious with that work because there are many features of the microbiota of the human gut that are only just being understood for their implications for well-being. And so that's not an experiment you would leap to do, but I do believe it's down the line. People have realized that even when we take antibiotics, possibly when we take drinks or we change our diet, we may actually have other health consequences other than just dietary of change in our microbiota. Now, it is possible to repopulate your microbiota, and so you could kill with Bdellovibrio and repopulate with the good guys, but it is important to bear that in mind in treatments. So that's an interesting question.
Thank you. The next question is from Pankaj, who actually, this kind of links into what you were saying, but his question says, can I use this method of induction in rodents?
Yes, it could be used there. We have only studied it in zebrafish, but the pathways would not be different. And indeed, we can map those kinds of genes that are induced in the fish in other animals. So those genes are conserved from fish to other animals, if that is the question. So yes, that is true. And part of the reason that we have done the work in the fish is to actually learn a lot more about the processes in higher animals.
Okay, thank you.
Thank you.
Another question. Can Bdellovibrio kill Microbacterium tuberculosis?
No, sadly not. So although that's an interesting gram-positive bacterium with an extra outer membrane, the chemistry of the outer membrane is different. So the Bdellovibrio does not recognize that outer membrane of Microbacterium tuberculosis, which is a sort of gram-positive bacterium with an unusual membrane. So yes.
Okay. Another question from Pamela. You may have sort of already answered this before, but this is more of the speed of things. So how rapidly do the pathogenic bacteria develop resistance to Bdellovibrio?
Indeed, they don't develop resistance to Bdellovibrio in terms of a gain or loss of function. Bacteria could acquire new abilities to coat themselves in a new outer surface, but to do that, they would have to acquire genes from an external source. So there is nothing that a bacterium can lose to become resistant to Bdellovibrio, because it's their very outer surfaces that are complexly recognized by the Bdellovibrio. So there isn't a single receptor that Bdellovibrio latches onto. It is testing the environment with several different receptors at once. And therefore, to lose all of them would be to lose the outer surface of the bacterium. And this is what makes this a more unusual, but actually more resilient means of killing bacteria.
Okay, thank you.
Thank you.
Another question from Pankaj, who says, how can Bdellovibrio co-work with human white blood cells?
I need to find out in, in our next research, but it can work side by side with human white blood cells, certainly by stimulating their actions when a pathogen was previously being ignored, but I don't yet know the pathways. That's what we're actually working to find out now. We are deciphering that at a gene expression level at the moment, but there's a lot more research in that. The reason we are doing that is to inform tests that we can do in higher animals. So maybe I haven't quite answered your question, but that we are doing that work now. We will publish it soon.
Great.
Just
well,
related
thank
to
you.
that, if I could just butt in.
Yeah.
So, to what extent can you detect immunity to the Bdellovibrio? If they're engaging with the immune system, they must be stimulating it to some degree.
Yeah. So we've looked in healthy humans, and we just sampled 100 healthy human sera, and we only saw very low levels of anyone that had, I think, just a few individual humans, like 2 or 3 humans out of 100, had any antibodies at all to Bdellovibrio. So it's not something that is commonly seen in individuals to have that, but obviously we have bigger studies to do on that. Antibodies to anything microbial that is new will be developed. This is why we call this a fire extinguisher, to use a phrase by John Rex, rather than a repeated treatment. This would be, yeah, it's a kind of, yeah, it is. It's got that element where you get treated once, and that should mop out your infection and take it away. This isn't a repeated pill that you take over a number of weeks.
No.
But it's different,
Yeah, thank you.
Yeah.
Thank you.
Thank you.
Cool. Brian.
Yeah, so maybe just one more question, if that's okay.
Thank you.
So you've talked about Bdellovibrio in the gut and how it affects the human body, but if this was to be kind of injected, and how would this then be released from the human body once it's done its job, in effect, if that makes sense?
Yes, it does. So one of the striking things has been that these bacteria do not last very long inside an animal body, but they're alive for a reasonable period of time. They are not strict anaerobes, and so in many environments like the gut, they run out of oxygen, but they're able to do one round of bacterial killing before they die, and therefore they're quite self-limiting. So the macrophages initially ignore the Bdellovibrio, but eventually kill it. And so 48 hours later, the Bdellovibrio are gone, but they have remained alive long enough to kill any pathogens that are present. So this is a kind of host-limited situation, in that the Bdellovibrio do die off in the host. They just don't die so quickly that they aren't around to have an effect. And that was the beautiful surprise, really, of the work we did with Serge. We hoped that they would stay alive, but actually they stayed alive plenty long enough to have a big effect on the pathogens that were present. And of course, if there were no pathogens present, they died relatively quickly. They stayed a bit longer if pathogens were present, because they could replicate on them for a while, but still be killed off by the immune system. So it's not that they die ever. They do die. They are killed. It's just that they persist for long enough to be predatory and have an effect, which is great and a good surprise.
Wow, that's really interesting, and that adds on to what you were saying about it being a fire extinguisher, and it doesn't spread around the body either. It doesn't circulate in the bloodstream. So if there is another localized infection, you have to retreat with it again, but for a separate infection. The one localized infection is one injection, and then you move on like that.
That's right. Thank you, Bryony. That's fantastic summary.
Thank you very much, Liz. I'll pass back over to Peter now.
Thank you.
Okay, well, thank you very much indeed, Liz. I mean, that was a fascinating talk, and nature never ceases to surprise us, does it?
I know, it's a great privilege to work with nature in the tiny as well as the large. they're our pharmaceutical industry of the future, and you know, they really know what they're doing. We just have to learn from them. The idea that a human can learn from bacteria that have no brains but are great pharmaceutical agents is amazing, really.
Maybe having a brain isn't necessarily a good idea.
Yeah, that's right. Evolution's the driver you need, really. It's,
Absolutely, absolutely. Yeah. Well, and on behalf of everybody that's watched you today and listened to you today, thank you very much for such a fascinating talk. And thank you for your contribution to SCI's talk series. It's been great.
Thank you very much.
Okay,
and
so
it's really good to meet you all.
So thank you very much for joining our webinar. If you don't know much about SCI, please go to our website, and if you're interested in joining, you can. All the details are here and on our website. And we look forward to meeting you more in future webinars. So thank you very much and goodbye.